Hello, everyone. My name is Serena Melissus, and today I'll be talking about stem cells and why they are such an important topic in biology. I picked this topic because I think it's really interesting that one cell can turn into so many different types of cells in the body. Before this project, I didn't know this in depth about stem cells, but after reading these articles, I realized how much potential they have. In this presentation, I'll go over one review article, two primary research studies, and then I'll finish by talking about a future experiment I would like to see done. Stem cells are special because they can become many different types of cells in the body. This process is called differentiation. Because they can change into different cells, scientists believe stem cells could someday repair damaged tissues, replace injured cells, and maybe even treat diseases that don't have good treatments right now. The review article by Zawaski and his team explains the history of stem cell research, where stem cells are already being used, and how they might help in the future. After reading this article, I understood why stem cells are one of the biggest areas of medical research today. They have the potential to change the way doctors treat injuries and diseases. Overall, my presentation shows why stem cells are such an important area of research and how they could improve future medical treatments. This reviewed article is called Stem Cells, Past, Present, and Future. Unlike the primary research article, this review didn't do new experiments. Instead, it looked at many different studies and explained what scientists already know about stem cells. One thing I learned is that stem cells can keep dividing while still staying stem cells. Then, when they are needed, they can develop into specialized cells like muscle cells, nerve cells, or blood cells. The article also explains that stem cells are already being used in some medical treatments, like bone marrow transplants, and research hope that eventually can repair damaged organs and tissues. Overall, this review gave me a good background before reading the two research articles. The first primary research study was done by Katashi Takashi and Shinya Yamanaka in 2006. Before this study, scientists mainly relied on embryonic stem cells. That caused concerns because they came from embryos. The researchers wanted to know if regular adult stem cells could be changed back into stem cells instead. They collected mouse skin cells called fibroblasts and used viruses to insert different genes into those cells. After testing a lot of combinations, they discovered that only four genes, OCT34 SOX2, KFL4, and CMYC, which is also the OSKM, were needed to reprogram the adult cells. This was a really important discovery because it showed that adult cells aren't permanently stuck being one type of cell. The results were very successful. The adult mouse cells began acting like embryonic stem cells. They formed colonies that looked like stem cells and could develop into many different cell types. These new cells became known as induced pluripotent stem cells, which are also known as iPSCs. One reason the discovery was so important is because scientists could now create stem cells without using embryos. Today, iPSCs are still used in research to study diseases, test new medicine, and learn more about how cells grow and develop. I think this was probably the most interesting study I read because it completely changed stem cell research. Now, the second research study was published in 1998 by James Thompson and his research team. Their goal was to see if human embryonic stem cells could be grown in a lab. The embryos they used were leftover embryos from IFV treatments that have been donated for research. The researchers took cells from the inner cell of blastocytes, which are the early stage embryos. Then carefully they grew those cells in special and specific lab conditions. Therefore, they watched to see if they would keep dividing while staying as stem cells. And the researchers were successful. The stem cells continued dividing for long periods of time without being specialized. They also created stable stem cell lines that other scientists could continue using for their research. This was the first time anyone had successfully grown human embryonic stem cells in the lab. Because of this discovery, scientists got a new way to study human development, diseases, and possible future treatments. Even though this research raised questions because embryos were used, it became one of the most important discoveries in stem cell biology. After reading these studies, I started wondering if stem cells could help repair damaged nerves. Nerve injuries are often very difficult to treat, so I thought this would be a good question for future research. In my experiment, I would use 20 mice with the same type of nerve injury. 10 mice would get stem cell treatment, and the other 10 would not. Over several weeks, I would measure how well the mice recovered by looking at their movement and how much nerve function returned. Using the two groups would make it easier to compare the results. For this experiment, my independent variable would be whether the mice received stem cell treatment. My dependent variable would be how much nerve recovery each mouse showed. The control variables would include mice of the same age, giving the same injury, feeding them the same diet, keeping them in the same living conditions, and observing them for the same amount of time. My hypothesis is that the mice receiving stem cell treatment would recover more nerve function than the mice that did not receive the treatment. If both groups about the same recovery, I would then reject my hypothesis. But if they treated mice recovered better, then I would fail to reject my hypothesis, meaning the results supported my prediction. I think this kind of research could eventually help scientists develop better treatments for people with nerve injuries. These are the 3 articles I used throughout my project. The first was my review article that explained stem cells and their history. The other 2 were primary research studies that showed important discoveries in stem cell science. Reading these articles helped me understand how stem cell research has changed over time and why it's still an exciting field today. Thank you for listening to my presentation. I hope you learned a little more about stem cells and why scientists believe they could play such a big role in the future of medicine.